Yo, yo, yo. What's up, everybody? Thanks for coming to Bioengineering 102. I think this is our second to last class. Um, but since most of the work here is hands-on, you might still be going even after the class is over. No worries. We'll still be around to answer your questions and do our best to help. I'm doing pretty okay. You know how it goes. Keeping busy. Um, Today, what we are going to discuss is how to create order custom DNA sequences. And so, during this class, what you should be able to figure out is how to read plasmids and rearrange plasmids and use restriction digests and ligations and stuff like that, transformations. But the question comes, what if you want a custom gene in a plasmid? What do you do then? And I had a priming issue and they did a straight repeat. What is that? Oh, okay. So if you're sending something off for sequencing and it said you had a priming issue, they'll just automatically repeat it. Usually, if you have a priming issue, it means that you don't have the correct plasmid or the concentration of the plasmid wasn't high enough. So make sure A, you ran the plasmid out on the gel and you saw a band, and B, that you selected the correct primer when you ordered the sequencing, and that you didn't mix up your samples. So those are the things that you need to figure out. So it's just one of the many ways sequencing. Usually when your sequencing goes wrong, companies will repeat your sequencing for free or cheap, usually free. So um, yeah, it's a good policy. I like it. But hey, Kent, how's it going, man? Today we're gonna discuss new putting new genes into plasmids and stuff like that. So generally, most of my work, I work with stuff that other people have already done, and generally when you're doing that, let me share my screen. Let's see. Let's see if we can just do a window capture. All right. So generally, what I do is I use scholar.google.com. That's the first place to start. Whenever you're working with genetic engineering, search the literature, see what's already been done, see what people will have found out. Since a lot of the stuff I do is with, human cell, you could see right here, it already given me recommendations. Um, so what we can look for is generally, if you're using something that's on human cells or something like that, you want to look for clinical trials or double-blind trials. So, for instance, if we're looking for gene therapy, let's say diabetes, um, let's try a clinical trial. Double-blind, randomized clinical trial. Yep. So here you can see that they used vascular endothelial growth factor, VEGF, in a double-blind randomized clinical trial. Um, And let's see what the results were. Gosh darn it. So many pop-ups and stuff. So they say it didn't work. So we know don't try VEGF when you're looking to do some diabetes clinical trials. Sorry about that, my throat. Here's one for treating heart disease and diabetes using VEGF. And they say not much happened also. Anyway, it's a good place to start. Some of the things in muscular dystrophy and gene therapy. You know, there's a lot of stuff working in muscular dystrophy and gene therapy. What you're really trying to find is you're trying to find what genes of interest will work for you, right? Now, if you need help getting access to a paper, you can always use sci-hub.tw, I think it is right now. Yeah, and sci-hub.tw. Oh wait, is this not? You guys aren't seeing any of this. Gosh darn it. Sorry about that. sci-hub.tw. Yeah, I wanted to share my screen. Sorry about that. So Sci-Hub, and you also, scholar.google.com if you want to look at this stuff. It'll help you be able to find different things. You always start with background research, right? You want to know what other people have tried and what's out there. You don't want to try something somebody else has tried and you know it's going to fail. Like, that's crazy. So the best thing to do is do some background research first. Figure out what you're going to do. The next step is you need to figure out the gene sequence of what you want. So say I want to insert a gene for, I don't know, what are people into these days? Living longer, bullshit like that. Anyway, you want to go to the NCBI. So if you just basically type NCBI into your search browser, and it should get you a link to the National Center for Biotechnology Information, the NCBI. This is where you're going to find all the stuff you need. What we are going to look at is protein. Even though we're trying to get a DNA sequence here because we want to, create a genetic engineering for our organism in some way, we want to get the protein sequence. Remember, we talked about the protein is really what's important. The DNA sequence is basically just telling us what protein to make, and then the protein does all the hard work. So for us, the protein sequence is the most important, not the DNA in this case at all. So we want to look at protein sequences. And for protein sequences, let's see, what do we want to do? What do we want to change? Do we want to change our eye color? I get a lot of people asking me about eye color. So let's see, eye color gene. What's the eye color gene? OCA2, wait, HERC2. Okay, say we want to overexpress genes in the eyes of an animal or something, so we could give it a specific eye color. So the genetic determination of eye color is between a couple genes. But it looks like OCA2 is pretty important. So let's look at OCA2. This is just Wikipedia. I'm just looking if we want to overexpress OCA2, if that does something. All right. So let's say OCA2, it seems if we use a bunch of it, what it'll do is it'll create more color in the eye, darker color. So let's look up OCA2. Homo sapiens melanosomal transmembrane protein. OCA2. All right. So now we got links. It looks like it's a pretty big protein. 838 amino acids, and it's got different isoforms. Oof, this is going to be complicated. But, well, does it say in the OCA2 what isoforms are the most important? Does not appear to say which isoforms. Isoform 2, 814. And then, what is this? Isoform 1, the 838 1 or whatever. Yeah. All right. We'll just assume 838 is the correct isoform we're looking for. And what we could do is we could click on, we want the FASTA sequence. Now that's just a sequence file display format, which makes it easier to look at. So here is our amino acid sequence. Starts with methionine. Doesn't have a stop codon on the end, but we can always add that in. Always remember the stop codon. Whenever I'm putting new genes in plasmids and stuff like that, you always forget stupid stuff like the stop codon and things. So always have stop. It never hurts to have an extra stop codon. Never hurts to have an extra stop codon. So it's okay. So let's copy and paste this stuff. Open up a notepad. And let's copy and paste the amino acid sequence. All right. So we got that. We got the OCA2 amino acid sequence. Now, the next thing we need to do is we need to codon optimize this amino acid sequence. Now, what is codon optimization? Does it even matter? Now, codon optimization is each and every organism has specific patterns of codon usages. You remember how codon. So you remember how some amino acids are coded for by different codons? So some of them have multiple codons. So let's look at this. All right, here we go. So because this is RNA, they have U instead of T. Generally, I like when they have T better, but let's have U here, whatever. So if you look at something like alanine, GCU, GCC, GCA, GCG, now the usage of these codons in different organisms is different. So what codon optimization does basically is it picks the most optimal codons for the gene that you're trying to make, right? We don't want the naturally occurring codons. They might suck or something like that. We want to have the most optimal set of codons. Now, there's some caveats here, right? Like, codon optimization is not 100% accurate. It doesn't always work. There are a lot of context to these things. So when you're trying to express a protein or something like that, there's a whole lot of things going on. While codon usage might be one of them, right? You might want to use an AAA or an AAG for lysine, but not for glycine. Everything in the system is working to keep this on its course and to continue to go. So a lot of things can go wrong in the process. A lot of things can affect the speed or the amount of protein being made, but it's a good thing to do. So how do we do codon optimization? It doesn't hurt. Codon optimization tool. Codon. All right. Let's try GenScript's GenSmart codon optimization tool. Of course, it wants me to log in. Why is my computer running so slow? Do I got a virus? All right, now we choose a host organism. Remember how we talked about how, why does it have a whole thing? This is really complex. So our gene is OCA2. The original sequence is that. Oh gosh, no, don't do that. Oh, no. All right, let's try this again. Now we're going to have to go back to the notepad. And we're going to have to remove all this bullshit. All right, let's try this again. Copy and paste in there. Yes, 838 amino acids, protein, exactly what we wanted. So expression host, we want humans. Though, whatever you're making, you can choose for. If you're making it in yeast and bacteria, yeah. If you find other websites, you can probably find um, other organisms that they'll even optimize for. So it's running. Oh gosh. Is it ever not going to run? Stop running. It's going to stop running. I think this might take forever. Oh, maybe not. Maybe it worked. All right, here we go. Now it gives us the details of our optimized sequence. You can even order it directly from GenScript. Click to copy the sequence. Boom. There is our nucleotide sequence. Now, if you remember, we talked about it earlier. There's one thing that we don't have that we need to add, and that's the stop codon. So, TGA, that's a stop codon, right? TGA, yes. TAG and TAA. All right. So we added in the stop codon. We got the sequence. The next thing is we need to insert it into a plasmid. Now, if you're doing this yourself, you'd find some general purpose plasmid, right? So you'd see, what do I want? So if we want a mammalian plasmid, probably one of the most common mammalian plasmids, pcDNA, 3.1, 3 point something. Now, why it's considered a mammalian plasmid is because it has the proper promoters, terminators, and all that other stuff to make it work in mammalian cells, which, not all DNA has. So that's something that you really got to be thinking about. So pcDNA, it's a plasmid map. We can get it from add gene. So that means I think we can get it in snap gene, right? So all we do is this. Let's go to new. No, let's go to import. And add gene plasmids. pcDNA 3.1. The original. Now, add gene is a place that distributes plasmids. You can't really get plasmids from them unless you're a non-for-profit. If you're a university, you kind of can, but really, it's just nonprofits and universities who get access to these. So let's see if I could find a non-tainted pcDNA 3.1. They all have all this stuff in them. Lots and lots of stuff. just look on the website where we were looking. Does it have like a vector database? Let's see, 2093. Let's see if that works. 2093. No. Why can't we get PCDNA in here without all this crap? All right. So the next best thing is just finding PCDNA with some other genes in it that we can just get removed. All right, this plasmid is not highly annotated at all. Ugh. Is that really all you have? Let's see. Detect some common features. There we go. Wait. Oh. Dang it. Come on. Add gene. Just let me download plasmid backbone. Import. Let's try SnapGene. Let's see if they have it. PCDNA. Here we go. PCDNA 3.1. All right. We found it. Yay. All right. So now we look at our plasmid. We see, where is stuff being made? Generally, you can look for the MCS, right? The MCS is the multiple cloning site. That's generally where genes go, the multiple cloning site, because that's where they put all the restriction enzymes. If you have all the restriction enzymes, then you could just basically use any MCS on any plasmid, right? So the next thing we need to do is we need to find out the MCS and what's there. All right. So we got our CMV promoter, which causes the transcription of the gene. The T7 promoter, which is for bacteria. And then we have all these restriction enzyme stuffs. So generally, what you want to do is you want to try to start near a start codon or something like that. Generally, NDE1 is a good one because it generally starts on it does start on a start codon, but it looks like we don't have any singular cutting NDE1s here. When it's black like this, it means it's single cut. So I think we have all the single cut sites. What we can always do is we can go up to enzymes, choose enzyme, type in NDE1. It looks like it is there. It is in our chosen enzymes. And we could go to enzymes and we could say NDE1. It says it only has one site, but it's nowhere near our MCS. So, oh well. NDE1. Gone but not forgotten. Oh yeah, there's an NDE1 site in the CMV. Okay, so what we'll use is, let's say, let's use Now, HindD3 is always on the other end. It's weird to see it there, but it's a pretty popular enzyme. Let's see, let's use HindD3 and XBA1, right? Oh, yeah, let's cut out a big chunk of this. All right, so XBA1. Here's what the restriction site looks like, TCT-AGA. So on the end of our gene, what we want is TCT-AGA. And then on the other end of our gene, if we're going to do HindD3, we need AAG, CTT. So on this side, we need AAG, CTC. All right. Now, if we're going to order the gene from a company and we're going to do restriction digest or PCR amplification and then insert it ourselves, the next thing we need to know is how many base pairs do we have as an overhang? Because the way restriction digests work is the DNA usually binds a little bit of ways from the restriction site, and then cuts it off. A good place to check is neb.com. NEB has all the restriction enzymes. They always have all the restriction enzymes. neb.com. neb.com. All right. So now what we're searching for is HindD3, 1, 2, 3. And we go to the restriction enzyme page. And what it should tell us is how many base pairs do we need at the end, minimally, to make this work. So let's see. HindD3. All right, what is going on here? This stuff used to be a lot easier to find. Product notes. There we go. All right. Star activity, properties and usage. I probably could just search this on the internet, right? All right, let's try the internet. HindD3, restriction, enzyme, base, overhang, restriction enzyme, base, what do you call it? All right, cleavage close to the end. So, as a general rule, 6 base pairs should be added to either side of the recognition site to increase cleavage efficiency, right? So some you could do less. If you're really, painstakingly trying to save money or save something, use this. But in general, 6 should be fine. Now, it doesn't really matter what 6 DNA bases you use. That's not really important. You just need anything. So what we can do is, let's see. We can go to our sequence. And so we have the restriction enzyme sites. So then what we also want to do is just add in 6 bases. So GCTGAC. And on this side, it doesn't matter, again, just 6 bases, GAATCG. All right, so we got our sequence now. So the important parts to remember: stop codon, right? So once you get your sequence from the NCBI, add in your stop codon. The next thing, if you're doing this yourself, find the plasmid, find the restriction sites you want to use. Then add 6 base pairs on afterward. All right? That's the secret. Now, once you got this, then you have to order it. Now, there's a lot of companies who do cloning and all this other stuff and will create and insert genes and stick them in plasmids for you. I tend to prefer GenScript because they really want to work with you, and they'll try anything. They'll try to put any gene in any plasmid. Where some companies are, they're like, oh, that gene has too many, repeats of G, 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 G. We don't want to synthesize it. It's a waste of our time. Whereas GenScript is like, if you want us to synthesize it, let's do it. So, let's see. Which service do we go to? We want... Is it under gene synthesis? All right, we want custom gene synthesis. All right, so here's the gene synthesis. Here's the turnaround time that you have, right? So we would probably do something like standard gene synthesis, trying to save money, Our gene is less than 8 kb because it's 3 amino acids for codon, and it was 838 amino acids, so that's about 2400, 2900 bases, which is less than 8,000. We should be all good. All right. So we already did our codon optimization here. Next thing we want to do is get a quote. This allows us to set everything up. Please input this stuff in a spreadsheet. All right. Why do we want a spreadsheet? It seems... Need to look for the old interface. I like the old interface. What's up with this? I don't want a spreadsheet. All right. So, gene name, we'll say OCA2. And then let's paste in our gene sequence that we made, that we found, and then made. Oh, come on, what's going on, browser? Oh, there we go. Is this website crashing on me? Come on, Firefox. Why are you crashing on me? Good Lord. All right, stop the madness. All right, there we go. All right. So we have everything in there. We do standard service. No, we already codon optimized. Now we can choose vector that we want to put our stuff in. God damn, the website's still not working? What's going on? Okay, so these just have simple cloning vectors. What we want, we'll do express cloning because we have all this stuff here. And shit, look, they have PCDNA 3.1. We have our restriction sites already, right? So Hindi 3 we chose, and what was the other one we chose? Hindi 3 and was it BAMH1 or EcoR1? TCT-AGA. TCT. No, we chose XBA1. All right, XBA1 and Hindi 3. Hindi 3 is the 5' one. XBA1 is the end one. All right, now they can send you 4 micrograms of plasmid, which is enough for you to transform it and purify some of your own. Or you could also have them do a custom plasmid preparation, which is actually kind of nice. And it's generally not that expensive. And you can make enough so that you have enough for all your experiments you need. You know, you're talking, you could have them make 2 grams of this stuff. So you can choose that. Let's just do free of charge 4 micrograms. And that's it. We just want this gene, 4 micrograms. In our plasmid, PCDNA 3.1, click submit. Now, they're probably not going to show you the price. Usually what they do is they email you a quote within 24. Oh, they showed us the price. $938. Woo, that's expensive. So the reason why it's so expensive is because our OCA2 gene is so big, right? So our OCA2 gene is 883 amino acids, 838. So that's 3 nucleotides per amino acid. So we're paying about 30 cents per nucleotide for the gene synthesis and then insertion of it into the plasmid, which isn't too bad. It could be cheaper. Generally, I do stuff a lot smaller. Now, GenScript isn't the only company that does this, so if you're concerned about price, you can look around. There are a ton of companies that do cloning, and they'll put it in whatever plasmid. So look around, search the internet, look for plasmid cloning, and I'm sure you could find a less expensive price. Yeah, I'm sure this isn't the best one. All right. So now, generally, what I do is I just find the gene I want. You're still just seeing the Google. Oh, were you guys able to see that the whole time, or was it just on like the Google search page? I hope you guys are able to see all the stuff I did. If not. So, yeah, generally DNA synthesis companies, they say they own the rights to whatever that they make, but like none of them actually do. I think somebody just created a standard terms of use a long time ago and everybody reuses it because like everybody needs to order DNA synthesis from somewhere. So if everybody's ordering it and people are still using it and selling it and using it for commercial purposes, like it doesn't make any sense, right? It doesn't make any sense that that you can't use it for commercial purposes. Like they're not going to stop you. And if they do, like they're not going to be able to get anything out of it unless you're like a big time company or something like that. They'll send you a cease and desist and you just pretend like you're ordering it from somebody else and move on with your life. Yeah. But otherwise, all right. So basically, from my point of view, if you're trying to get your own custom gene and put it in a plasmid, just pay somebody else and move on with your life to do it. Like, don't take the time and effort to do it yourself. I know this class, we learned a lot about restriction enzymes and all this other stuff and how to do all that stuff. All that is so you know how everything works, right? And so you could do it if you need to. But if you don't need to, you don't want to do cloning because cloning is hard, is difficult, is time consuming, as I'm sure you're all learning. What you want to do is you want to be able to pay somebody to do this stuff for you, right? So like go and pay somebody else to do it. Find the price and have somebody else do the cloning. You're generally not paying that much for the cloning and stuff like that. You're paying everything for gene synthesis, and you're going to have to do that anyway, no matter what you do. So save yourself, the effort and the stress of trying to clone it yourself. But yeah, it's pretty easy. Anybody could do this shit. You can do it. You can order this stuff. You just have to know what to do. So in the future, if you want to order plasmids for, work in mammalian cells or work in animals or something like that, now you have a general idea of how you can do that. I hope everything's going well in the class. I know it's a tough one trying to do these experiments. There's definitely a learning curve for all this stuff. So don't feel bad if it's slow going because doing lab work it's like learning a guitar or a musical instrument. Like there's things you have to learn that you just figure out what works and what doesn't work and what incubation times and temperatures work and what doesn't work and all this stuff. I got GFP, P-Dawn, and P-Dawn again. Dope. So Tom, how long do the bacterial stabs last typically? If your bacterial stabs are stored in the fridge, they can last months. What does months mean? 3, 6 months, it really depends. So P-Dawn, only a single band in the gel. So when you added AAV, when you added the restriction enzymes to P-Dawn, are you saying they didn't cut at all? Kent, Are some companies less willing to manufacture plasmids for particular organisms? No, most companies don't really care. They'll manufacture plasmids for anything. It just more depends on, the like, if you're trying to manufacture a plasmid for like Drosophila or something, it's going to be a lot harder. Um, yeah. Hey, AAV, I would say post your questions in the Google Classroom. If you have already, I'll check them out. Sorry if I haven't, with the exact stuff that you did, because then it makes it easier for me to answer questions, having all the information in front of me, and you also writing it down so we can see what's going on. Sound good? Okay. But, it's good to just post questions and problems anyway, because it might be something else and I can help you troubleshoot it. We can. All right. So keep up the good work. Hope you all are making progress. And we will speak again next week. I think next week is our last class. And yeah, bring your questions. Try to get the experiments done and bring your questions so that we can help figure out what's going on and get you to get your experiment to work so that you can finish up the class. All right. I will see you next week, and I hope everything is going well.